Professor Das did postdoctoral research work at University of Utah, USA. He was also associated as MNRE Renewable Energy Chair Professor for 3 years and Professor for more than 32 years at IIT Kharagpur. His commendable contribution towards development of commercially competitive and environmentally benign bioprocess for the hydrogen production. His recent work on biohydrogen process for the maximization of gaseous energy recovery from the organic waste is worth mentioning. He has more than 170 research publications in the peer-reviewed journals and contributed more than 38 chapters in the book, authored 6 books and edited 2. He was awarded IAHE Akira Mistyu Award 2008 and BRSI Malviya Memorial Award 2013 for his contribution in hydrogen research. Professor Das, now I would request you to take over the session.
Hello, I want to share my presentation. That is the thing.
Sir, you yes, sir. Sir, you need to click on your presentation and then click on the slide mode.
No, I could not share this because
Sir, the meeting screen where you have logged in, there you can see an open share tray with upward head arrow. You need to click on that.
where that at the top. No,
No, the meeting that you have joined, next to the mic mute button, you will see an upward head arrow. You need
that
that.
that I click. That I know,
Yes,
but
sir. Open share tray.
open share tray. I have
Yes,
done that,
sir.
but this
Then
not coming.
entire screen.
Then enter screen. Okay,
Yes, sir.
let me check.
Mhm.
Nothing is coming actually.
Sir, you had opened your presentation in a separate window.
Yes, I have already opened.
Okay, okay. Now, sir, cancel that open share tray. Will start again because earlier your screen was being displayed, but now it's not.
Oh, I see.
Mhm. So, can you access it?
I will try to make it, but I am already here.
So, where?
Share button is on actually. Open share. I have done that.
Yes, sir. Sir, just sir, ma'am will share your PPT. Will that be
Okay,
okay?
okay, okay. That will be okay.
Okay,
Only that is the PDF file. That is no problem.
okay, okay, sir. Ma'am
It
is
will be
sharing.
better
Hold
if I can share by myself because I can control that. But
on.
anyhow, if problem remains, that I have nothing to do. Okay, that's fine.
Yes, sir.
Now
Sir,
it is
is the
okay.
yes, sir. Yes, sir. You can continue.
Okay, so you change the slide. Okay,
Definitely, sir.
okay, okay,
Just let
okay,
us know when to change the
okay,
slide.
okay. No problem. No, absolutely no problem. Very good afternoon to all of you. That and uh, Welcome back to this auto workshop that program that today the topic I'm going to cover, that is the scale up biofuel production processes from organic waste. So before I start my presentation, I want to share I want to share the thought of Albert Einstein, that what he has said, he told, the imagination is more important than knowledge. The for knowledge is limited to all we know, we now know and understand, while imagination embraces the entire world, and all there over ever will be know and understand. So that. So if you want to, in short, what I want to point out, that if you want to do something good, so you have to imagine something. Imagine that that I want to do this and this. Then and only then you can reach to your target, because that is very important. What Albert Einstein has pointed out. So, in my kind introduction, it was pointed out that I am associated as a INE AICT Distinguished Visiting Professor at SRM Institute of Science and Technology, Chennai, and Heritage Institute of Technology, Kolkata. Previously, I was the professor and head and eminary chair professor at IIT Kharagpur. At the bottom, I have given you the my website link, that is www.biowaste2iitkgp.in. So anybody has any interest on our research work, please visit our website so to get the information in details. May I have the next slide, please? So the question that has come to us, to our mind, that what do you mean by biofuel? Any fuel that is produced from organic matter that usually form living or living material in a short period of time, days, week, even month, is considered as biofuel. That means what I want to point out, that anything is coming from the living things, any fuel we produce from the living material, we consider as biofuel. Now, as you know that we have a lot of biomass. Biomass means we have lot of agricultural product, that is also biomass. Now, this biomass can be organic mass, can be biological converted into the fuel, both gaseous and liquid fuel. Also, chemically also it can be converted. So, since this biomass is converted to this fuel, that is why, in a broader sense, the fuel that is generated, we call it biofuel. But but our process is purely biofuel, because all through we are using the biological system. The Our raw material coming from from the living system also, we are using the living system for converting the biomass into fuels. May I have the next slide, please? So the question comes that why we need the biofuel. The reason is that the sustainable biofuel are essential to ensure constant ensured supply of energy for individual and industry. Advanced biofuels will reduce our dependency on the fossil fuel and limit our impact on the environment. It is also argued that the investing in biofuel production may boost the economy of the developing country. Now, here I want to just point out that that as per that biofuel production from the is concerned, this is more in the developing country rather than developed country. Because if you see the worldwide scenario, that higher percentage of biofuel is used in the developing country as compared to developed country. May I have the next slide, please? So this is the renewable energy sources we know. We have solar energy, we have wind energy, we have this tidal energy, nuclear energy, we have bioenergy. So the different renewable energy sources we have. Now, now here, I shall just next slide. I shall pointed out the potentiality of the solar energy. Tremendous potentiality we have. Whatever a fraction of solar energy actually we required to meet the energy demand of the world. So that the question comes the how fruitfully we can utilize this solar energy for the energy generation process. So there are several avenues are there. Now this the bioenergy is more attractive. The reason is that it is less energy intensive and less it contributes less pollution to the environment. May I have the next slide? So these are the different sources that we have that you can see that we have renewable that no, the non the screen is so small. So that is why PPT is very much useful for me. Non-variable is there and another is the variables are there. So so these are the variables we have solar and wind, that is variables. But if you look at the solar and the fossil fuel, that is the fixed energy, non-renewable energy. And non-renewable that non-variable renewable energy is given in the upper side. You can see it here. Now, what I was talking about, that potentiality of the solar energy. You see, the solar energy we get per year about 23,000 terawatt. And what is our requirement? You see that our requirement is very less. As per our requirement, the screening base is very small, actually, small screen. Can you make the screen little bit bigger? Okay. So, a fraction, what I wanted to tell, the fraction of the energy is good enough to meet our the energy requirement we have in the world. So now here you come that what is the importance of the renewable energy. Now here you see that biofuel, that requirement now at the present situation is about 10% as compared to other fuel like nuclear, coal, and other fossil fuels that we have. Now, now if you look at because another slide I could not you cannot see because it is overlap with other slide. This actually, that is, if you see that energy demand, that will increase like anything, because we know that China and India are the vastly populated country. So since we are staying in a vastly populated country, our energy requirement will increase with respect to times. And you see, 2000 by 2035, China required huge amount of energy, and then come the India, then other countries that we have. May I have the next slide? Now, what is the current scenario? The current scenario is that fossil fuel, in the morning I was telling, the fossil fuel provides almost 80% of the world energy supply. That is, and 20% comes from the other sources. Out of this, oil provides 40% of the total global energy need, and fuels 90%, because out of this oil, this energy that we have, the 90% of this oil energy that used in the transportation sector. At the current rate, the consumption will be running out of petroleum for 50 years, natural gas 65 years, and coal over to 200 years. This I pointed out in the morning. Now this is this is the importance of the biofuel. You see, the first important it is biodegradable, stability, that and reduces the dependency of the petroleum fuel, and environmentally acceptability. Now, biofuel may be available in liquid gaseous form as well as in the liquid form. That also, the biogas, we have the biosynthesis, land, plant, and sea algae that is used, and biotransportation also. This can be utilized. So it produces carbon dioxide. This carbon dioxide produces biomass. From the biomass, we can produce biogas and biofuels. This is how the system, this cycle runs. May I have the next slide? So this is the scenario that we have. Biofuel, we have different forms. We have biohydrogen and biomethane. Biomethane is largely used. We have several anaerobic digester, and which produces methane, and that is successfully utilized throughout the world. And this is used for the production of CNG. CNG means compressed natural gas. Because what is the use of CNG? Because, as you know, it is the methane is the lowest hydrocarbon, and when it burns, there is a less possibility of producing the particulate matters. Now, you might be aware that in Delhi there is fog formations, and due to this burning of the fuels, and this fog formation was controlled by replacing this bio that diesel and petrol by CNG, compressed natural gas. Because when compressed natural gas burn, methane burns, it doesn't produce any kind of particulate matter. Because when higher hydrocarbon burns, it produces some kind of particulate matter, because all carbon cannot be burned out. It's a portion of carbon will be remain in the form of particulate matter, and that causes the fog formations. And another very interesting issue that we have, that I'm going to share today, that where we have lot of interest, that is the biohydrogen production, where little attention has been given. Now, besides that, biofuel can be available in the liquid form also. We have bioethanol. Largely ethanol is used, and as you know that it is the mandate of the government of India that we shall have to use about 10% of ethanol in the petrol. So 10% the gasoline is to be replaced by the ethanol, and so that reduces the petrol consumption in our country, because, as you know, that petroleum oil, the petroleum available in India is only for 30 years, even less than 30 years. So naturally, that most of the petroleum, that crude petrol, that petroleum that we import from outside, and so we spent a lot of foreign money for purchasing the petrols, so for purchasing the petrol. So that is one of the biggest issue. Another thing is that what you call a green fuel. Green fuel means that is the conversion of bio waste or the organic waste into electricity. But the electricity doesn't cause any kind of pollution problem. That is called green fuel. So these are the, as far biofuel is concerned, we have three different things. One is we have gaseous fuel, we have this liquid fuel, and we have electricity also. And this can be produced from different organic matter, and this organic matter are usually produced from the biological sources. And this organic matter can be classified into two types. One you call solid waste, another we call liquid waste. Now solid waste, we have municipal waste, we have agricultural waste, we have industrial solid waste. And there are different municipal solid waste. We have organic litters, household waste, and the sewage. And agricultural waste, we have algal biomass, energy crops, food crops, and lignocellulosic material. Industrial solid waste, well, food processing industrial waste, dairy industrial waste, and the agro industrial waste. Now, as for liquid waste concerned, we have industrial wastewater and domestic wastewater. Industrial wastewater, we have distillery effluent, paper mill effluent, starch wastewater, and pharmaceutical wastewater. And domestic wastewater, we have canteen waste, we have household wastewater, and the starchy wastewater. May I have the next slide? Now, question comes: what are the different this technology available for converting this organic waste into the energy? So in the biofuel sector, we have biohydrogen, we have biomethane, we have bioethanol, we have biobutanol, biodiesel, bio oil by pyrolysis, and liquid form or and liquid fuels. And gaseous gasification of the technology, that is chemical gases, thermochemical gasification technology, that includes the hydrothermal gasification, gas by plasma pyrolysis, and syngas by production pyrolysis. Another is the incineration technology, the augmentation in thermal power station, and heat generation for the household uses. May I have the next slide? So, as per the fuel generation is biofuel generation is considered that it can be produced four different types of organic material, and first we call the first generation biofuel. First generation biofuel, it is the based on the fermentation of food-based biomass, that is the biofuel which is derived from the sources like starch, sugar, corn, wheat, sugar beet, cassava, animal fat, and the vegetable oil. So that what I want to mention, that here the problem is the food versus energy. So if your country give importance to the food, then we will we will mostly concerned about the food production, because since we are in the vastly populated country, we naturally we are more interested about the food. The food versus oil, that conflict is will be there in case of first generation gaseous fuel. Now this can be overcome if we use the second generation biofuel. In the second generation biofuel is produced from the different type of biomass. A biomass wide ranging term, meaning that sources from the renewable rapidly as part of carbon cycle. So it is a complex biomass, and this means it is This means that it is it is difficult to release it from the cell wall to break down and the cellulose into the sugar and through the microbial fermentation. What I want to mean, in case of second generation biofuel, mostly it is produced from the lignocellulosic material. And major bottleneck of the lignocellulosic material is that lignin is embedded on the surface of cellulose and hemicellulose. So that is, and as you know, that lignin is a very complex material, complex organic, and very difficult to degrade. So naturally, you have to remove that lignin material before your enzyme cellulases that may act on the cellulose and hemicellulose material. So that is the major bottleneck. So pre-treatment is very much required for utilizing this lignocellulosic material, and this research still going on to make the process commercially viable. May I have the next slide? In the third generation biofuel, we have this is the various that lignocellulosic biomass that we have. You can see it visualized from this particular slide. So I have given this PPT to Professor Pandey. So if you want that he can share with you. May I have the next slide? So third generation biofuel, algae is considered as a third generation biofuel because it can produce, it can require very small amount of land for the production of this biomass, because you don't require in a huge area of land for the production of algae. That is why the algae plays very important role for the biofuel, and algae can be used as a source for the biodiesel production. Also, this algae can be used as a supplement for any kind of wastewater for the production of biofuel, and also algae can be converted into into the energy through the thermochemical process. So the merits of the algal-based biofuel with the microalgae have potentiality to produce 100 times more oil per acre than the terrestrial plants. Algae do not compete with the food crops for land, as they can grow on non-productive and non-arable land using a variety of water sources. The limitation is that development of the algal fuel is at an experimental stage, and production is currently very costly due to limited knowledge of scale up possibilities. So what I want to mention that that the algae production we can be taking place in two type of reactor. One is called the raceway plant. The raceway plant is exposed to the atmosphere, and the photo bioreactor, that is in the mostly in the tubular reactor in the control atmosphere. Now tubular reactor or photobioreactor, you can protect the contamination problem, but raceway plant, since it is exposed to the atmosphere, it is very difficult to control. Could you please go back to the previous slide? Go back to this slide. Please go back. That fourth generation biofuel is kind of genetically engineered to consume more carbon dioxide from the atmosphere. So we produce some crop which can produce, because as you know, the crop can use carbon dioxide from the atmosphere and it produces oxygen, and the carbon-rich biomass is then converted to fuel and gases with the synthesized microbes. So the merits is it uses the raw materials that essentially inexhaustible, cheap, and widely available. Limitation is that generation of designer bacteria with with new useful property required for revolutionary scientific breakthrough in several areas of fundamental research. Let me tell my experience that we have, because I I visited the Kelvin lab in California, and they have this project from British Petroleum, and the project is how fruitfully we can use the lignocellulosic material to biofuel. So now this research group has been working in different way. They're working not only with the process for biofuel production, but also they are concerned about the raw materials, how the raw materials can be improved. I can give the very typical example that when you talk about the degradation of lignocellulosic material, it contains the cellulose and hemicellulose also besides the lignin. So after removal of lignin, so you have to degrade the cellulose. Now cellulose, that is available in two different forms. One is called crystalline cellulose, another is amorphous cellulose. Now crystalline cellulose is very difficult to degrade, but amorphous cellulose is very easy to degrade. That is the point I want to mention. So now this research group, they are trying to develop some kind of enzyme which can degrade the crystalline cellulose very easily, so that the utilization of the cellulose will be very successful. That is one group. Another group is working that on the how we can produce less lignin containing the lignocellulosic material, and the different way they are working just to make the process more attractive. May I have the next slide? Now, I in the morning I was talking about the carbon dioxide that emitting. So 10 carbon dioxide emitting country has been shown here, and India stands in the third position. You can see in the particular figure in the right hand side. So, this problem is quite severe because China is the more severe, then USA, then we have India, then we have Russian that federation. Now it has been observed that 80 the about 68% energy that comes from this that agriculture contribution is 11%, industrial processes contribution is 7%, and the fossil fuel that is used in the transportation and other sector, that contributed in the 68%. The maximum carbon dioxide emitted from energy sector, that India is among top 10 carbon dioxide emitting country. May I have the next slide? So that carbon dioxide emission, that is the major issue that we have. May I have the next slide, please? Now, we are talking about the hydrogen, that why we are thinking for the hydrogen. And this is the trend of utilizing the fuel usage. You can see that if you look our civilization, that we started by using wood as a source of energy, then animal fat, then coal, then petroleum, then propane, then natural gas, then hydrogen, and finally we have the fuel cell. Now, so since it is stepping into hydrogen, the why it is hydrogen? Because hydrogen, when it burn, it produces only water. So no way it is polluting the atmosphere. And fuel cell, why fuel cell? Because in the fuel cell, hydrogen can be directly converted into electricity. There also there is no chance of pollution. So this is the trend that we have. So that is why hydrogen plays very important role in the years to come. May I have the next slide? May I have the next slide, please? So this is the carbon. The hydrogen is considered as a zero carbon fuel, because the zero carbon emission, by balancing the measuring the amount of carbon release with an equivalent amount of carbon sequestered or offset. It is mainly used for carbon dioxide emission process associated with transportation, energy production, industrial sector. So carbon neutrality concept is extended to include other greenhouse gases like methane, nitrous oxide, hydrofluorocarbons, and perfluorocarbons, and sulfur hexafluoride. May I have the next slide? And I'm glad to report you see that there is two countries, two states is there. They have zero carbon emission that we have. Worldwide, there is the two state, but there is the zero carbon emission is there. So, what is zero carbon fuel? Because it does not produce produce any carbon like monoxide, carbon dioxide on combustion. That is called zero carbon fuel. May I have the next slide? Now, this is the stoichiometry, how hydrogen performs the water, we know it. So this is how it reduces the pollution problem. May I have the next slide? This is the hydrogen economy that we have. You see that the industry, we have air pollution, we have the environmental destruction, we have the dependence that dependable, and we have other pollution fossil fuel that economy is there. So these all lead to carbon dioxide. Am I right? And hydrogen economy is something different. In the high form hydrogen, we get the energy, and we can do the electrolysis of water, and it produces the hydrogen and oxygen. And this hydrogen we can use as the energy in the transportation sector, or you can use in the combustible energy. Again, it produces water. Again, we electrolysis. This cycle can move forward. So nowadays, this plays very important in the hydrogen economy. I want to report here that hydrogen is considered highest that energy content as compared to any other fuel that available in the nature, that is contains 143 kilojoules per gram, near zero pollution, and potential production from the variety of renewable resources. This is the advantage we have. And at the top, right hand side corner, you can see that it has the blue flame, because if you see the flame of LPG or methane, it is yellowish flame, or when hydrogen burn, it gives the blue flame. Only the difference is that when hydrogen burn, it produces blue flame. Not only that, and it gives some kind of noise. Because why? Because it produces water, and water acts as a fire extinguisher. So you'll get always kind of noise in the flame. But in case of other fuel, that gaseous fuel, when it burns, you will not get any kind of noise. May I have the next slide? So this hydrogen can be produced by two different ways. One is called non-biological process, and another is the biological process. So raw materials, whatever raw materials is there, maybe in the non-biological process, we can use the biomass as the raw materials, because some of the non-biological process, but biological process we mostly use the biological material, and by using the different living system and enzymatic system, it can be produce this hydrogen. May I have the next slide? And this is the technology that is available with us. That is the thermal and thermochemical methods that is used, how it is produced for One is called cerium oxide two-step cycles, copper chloride hybrid cycles, and another is we have this water hydrolysis I told you, and the steam reforming and partial oxidation, and this is another process that we have. Plus, we have the biomass conversion process that we have by using steam. In presence of steam, in presence of air, it can be converted to the energy, hydrogen. May I have the next slide? So that that as per biological hydrogen production is concerned, that two type of organisms are mostly involved for producing this biohydrogen. That one is in presence of light, another is absence of light. Now it has been observed that photobiological system and is no good for hydrogen production the rate of hydrogen production is very less. And not only that, now if you look at the history behind the biohydrogen production, that scientists they've found out that algae has the capability for producing hydrogen from the water, and we know that two-thirds of the earth is water. So scientists thought that if we can convert water into hydrogen, we can solve our energy shortage problem. But when they try to utilize this water, they found that with hydrogen it produces some kind of molecular oxygen, and that gives some kind of inhibition effect on the hydrogen producing enzyme that is required in this system. So whole research work then directed that just to find out that oxygen tolerant culture. This research still going on, and we established that both algae and you have the photo fermentative organisms, whatever is there, they use for hydrogen production is no good because the rate of production is very less. And not only that, during the production process they produce some kind of pigment, and this pigment will keep some kind of shading effect, and that reduces the photosynthetic efficiency or light conversion efficiency of the process. But in case of other system, that is we call is dark fermentation process, it doesn't require any kind of light energy. It can grow in presence in absence of light energy, and that that system is more good because it produces hydrogen very fast. So we today will be giving more stress on this dark fermentation process. May I have the next slide? So this is the biological method I was talking about, the biophotolysis. You can see the green pigment formation, you can see. But here I told you that molecular oxygen that is produced here during using the algae, that gives a inhibitory effect on the hydrogenous enzyme that is responsible for the hydrogen production. But in case of photofermentative process, you can see here that that at the bottom, that in the red pigment formation is there, that gives some kind of shading effect. So initially you will find the rate of hydrogen production is okay, but as the process started, after some time is drastically reduced. So these two processes is no good for this. And right hand side we have microbial electrolysis cells, and the microbial electrolysis cells we can convert directly waste into the electricity or the waste into the hydrogen. But the problem is that it has the scale up problem because the stoichiometry's hydrogen production is quite attractive. But if you go for the higher system, the scale up of the system poses a lot of problem. But in the top, we have dark fermentation process. We can easily grow the organism in a reactor, and it produces hydrogen very nicely. May I have the next slide? One of the major advantages is that this process is usually work at ambient temperature and atmospheric pressure, so it is less energy intensive. Now, as I told you that hydrogen production, we have one is light dependent and the light independent. Light independent, we have dark fermentation, we have microbial electricity system. You will see that both this process is good. The reason is that that the hydrogen yield is almost same. So, but in the microbial electrolysis cells, the scaling up problem is there. So, large scale application we have problem. But in case of photobiological system, I say you see the yield is very high. But since there is the inhibition to the enzyme which are responsible for the hydrogen production, that reduces the hydrogen production drastically. That is why it is no good for hydrogen generation. And photofermentation process that also has a good stoichiometry, but it has the shedding effect. That is why it reduces the hydrogen. So these two processes are no good for the hydrogen production. But dark fermentation process appears to be the fuel process for the future for the biohydrogen production. May I have the next slide? May I have the next slide? This is actually dark fermentation means and it can grow in presence or absence of light, doesn't matter. And this is the stoichiometry we have. If glucose is burned in presence of oxygen, it produces 6 moles of carbon dioxide, 6 moles of water. When 1 mole of that 1 mole of this glucose converted to hydrogen, it follows the acetic acid pathway. It produces 4 moles of hydrogen and 2 moles of carbon dioxide. So what I told you that the biological process has less footprint, carbon footprint, because it produces less amount of carbon dioxide to the atmosphere. And if it produces the follow the butyric acid pathway, it produces 2 moles of hydrogen, so it reduces just half as compared to the acetic acid fermentation process. And then if it produces glucose is produced ethanol, it doesn't produce any kind of hydrogen. So this process is no good for the hydrogen production. May I have the next slide? Please, next slide, please. So the advantages of this that dark fermentation process is that it is efficient hydrogen production process compared to other biological process. As I pointed out, it leads to the formation of the other fuel like ethanol, acetate, and butyrate. So this acetate, butyrate, and ethanol, we can, if we pass it through the anaerobic digestion process, you can easily convert this to methane and carbon dioxide. So if you with total energy production you can increase if we use this biohydrogen production process followed by biomethanation process, what you call biohydrogen. And we consider the biohydrogen process will be the process that will remain with us in the years to come. But it does not require any kind of light energy. Scale up is comparatively easier and cost effective, and different waste material with a high organic load can be used for this hydrogen production. These are the advantages of the dark fermentation process. May I have the next slide, please? Now, this the dark fermentation process is usually carried out by the two different group of micro process. One is microflora, one is called mesophilic, another we call thermophilic. Mesophiles that usually grow in the range 20 to 45 degrees centigrade, mostly in the range of 35 to 37 degrees centigrade. Thermophiles usually grow in between 50 to 70 degrees centigrade. Now, thermophiles has some kind of positive effect because if we grow the organism at around 60 degrees centigrade, as you know, that most of the pathogens that will kill, and when you use any kind of waste material, there is every possibility of presence of different type of microbial contamination. Am I right? So it's naturally, if we go for the thermophiles, this contaminate this they will kill all the pathogenic organisms. So that microbial contamination problem greatly reduced in the thermophilic organism. But in the mesophile, you this problem you have to whenever you work with this problem you have to sort it out. So the organism that is used for mesophilic dark fermentation process is the Clostridium botulicum, Saccharomyces cerevisiae, the Enterobacter species, Klebsiella species, and Citobacter species. No, and we have in the in the thermophilic dark fermentation process different type of organism we use with thermo anaerobacterium species, then we have clostridium species, we have thermotoga species. So different type of organisms we use in case of thermophilic dark fermentation process. Now I want to add here that now you since we are living in the tropical country, our ambient temperature usually very close to the mesophilic temperature as compared to the thermophilic temperature. Only in case of summer, our ambient temperature increases 40 to 45 degrees centigrade. Now, now in case of we think that in case of distillery effluent, whose temperature is around 70 degrees centigrade, if we put it in a insulated reactor, we can easily maintain at 60 degrees centigrade. So no additional amount of energy you can put for the growth of the thermophiles. So in that for handling the distillery effluent for hydrogen generation is quite useful when we go for the thermophilic organism. May I have the next slide? Now mesophiles actually then these are the two types. One is called obligate anaerobe, and another is the facultative anaerobe. Obligate anaerobe, as you know, they cannot tolerate trace amount of dissolved oxygen concentration. Just a minute. So we have this is the bottleneck we have. If little oxygen contamination is there in the system, whole process will be affected. But this will not be affected in case of facultative organism, because facultative organism can tolerate trace amount of oxygen concentration, because in presence of oxygen they produce a cell mass, in absence of oxygen they produce hydrogen. So this process, the facultative process is easy to handle as compared to the obligatory anaerobe, and thermophile process is mostly their obligatory anaerobe. May I have the next? May I have the next slide? So this is the metabolic pathway that is followed by the facultative organism. I don't like to go in details. So you just to go only. I want to point it out that when we started research work, we started this biohydrogen research work in the year 1999, and then we observed that most of the research work has been concentrated on the photobiological system as compared to the dark fermentation process. So we started working on dark fermentation process, and we isolated one bacteria, or we call it a cluster, the Citrobacter. We call it the Enterobacter cloacae, and this Enterobacter cloacae produces hydrogen to a very, to a very high percentage of that organic matter that is converted into hydrogen. So we very little information was available in the literature regarding this organism and how it converted to hydrogen. Then we try to establish the fact that how this organism produces hydrogen, and we observe that this formate hydrogen lyase, this particular enzyme is responsible is present in the organism responsible for the high amount of hydrogen production. May I have the next slide? So this is the metabolic pathway of obligate hydrogen production. I told you here we use the kind of hydrogenous enzyme that we use we have ethanol, we have butyric acetic acid, we have butanol, butyric acid. So both acetate and butyric acid pathway we can produce hydrogen, but the ethanol and lactic acid fermentation pathway that we consume a lot of NADH, and this NADH they utilize for the production of ethanol and lactic acid. So if we the later on we try to close this pathway and try to develop some kind of mutual strength, how it can, so that we can save some NADH which can be utilized for the hydrogen generation. May I have the next slide? So this is the scale up that things that we have done. This is I want to share with you that we started with you can see the small bottle that uh, the serum bottle we use just to grow the culture, and then we use the cylindrical vessel. You can see it is a jacketed vessel just to control the temperature, because we pass the hot water through the jacket just to control the temperature, and we put it in a we put the magnetic flare at the bottom just to make the cells in suspension, because the cells are the insoluble mass. If you don't put the agitation, then cell will settle down, and then the reaction will take at the bottom part of the reactor. No reaction takes place in the upper part of the reactor. The hesitation is very much required. And then we use the photo fermentation process. This is the New Brunswick fermenter that we use. We use both the New Brunswick fermenter as well as the bioengineering AG fermenter. This is the control model we have. In the control model, we can control the different parameters here. And then we we use the 20 liters that custom made reactor that was designed by ourselves. And here we try to use the immobilized system, because in case of a suspended cell system, we find during the continuous operation of the process, the cells are wasting from the reactor. And if the rate of, we know in case of anaerobic fermentation process, the rate of growth of the cell is very less. So naturally, so you have to do some kind of recycle of the cells. So to avoid that kind of situation, we use some kind of immobilized system just to fix the cell on a solid matrix, so that we can pass your waste material through the reactor and directly it convert it to hydrogen and carbon dioxide. Then from this 20 liter we go to 500 liters, you can see, and from 500 liters we go to 10,000 liters. This is the 10,000 liter reactor that has been installed at IIT Kharagpur. If you visit that anytime, you can see that. This is how the small scale can be converted into large scale, and finally, the technology has been transferred to the Dhampur sugar mills for the commercialization of the process. May I have the next slide? This is a close view of the 20-liter reactors. I was custom made reactor. I told you that the operation of this video of the operation of this process is available in our website. If you visit our website, you can see that how we operate the system, how the hydrogen production take place. So everything is demonstrated there. So please see, this is a small video is there in the website. And when we use the one interesting thing, we try to find out that when we use the immobilized system, the immobilized system means you fix the cell on a solid matrix. So when the hydrogen production takes place, hydrogen is entrapped inside the solid matrix. So gas hold there is a gas holdup problem. As the gas holder problem is more, then what will happen that the working volume in the reactor that will be reduced. What is the working volume? Working volume is the liquid volume because the reaction take place only in the liquid phase, not in the neither in the solid phase or gaseous phase. So since the liquid volume is the we try to use different type of reactors, and we find the rhomboidal reactor, which is nothing but the convergent and divergent reactor. You can see it here, and that is used for removing this, for reducing the gas holder problem in the reactor. May I have the next slide? This is the closed value of the 50-liter reactor. You can see 50-liter reactors. Then we pass it through the 500-liter reactors. So this we successfully operated here this reactor, and this is how water, that gas we collected through the process of water displacement that we collected here. May I have the next slide? Another reactor we try to operate, that is the 800-liter reactors, but here we use the immobilized cell, not suspended cell. We use the immobilized cell. We use the coconut coir as a solid matrix, and we find the performance water of the reactor is quite good. May I have the next slide? This is the 10,000-liter reactor. You can see that at the corner you will find 10,000 liters, and besides we have two 3,000-liter reactors, and these reactors are basically we prepared our feed for the final reactors, because these reactors are used for preparing the feed for our this 10,000-liter reactor. So from that, this we pass our feeding material to the 10,000-liter reactor, and at the here you can see middle, it is stainless steel. You have 500-liter reactors. So this 500-liter reactor is used for the production of the cell mass, or the production of the inoculum, and then this initially we take it 4.5 cubic meter. That is then 4,500 liters of feed we take it here, and then we transferred 500 liters of culture here, and when the fermentation when the rate of hydrogen production is maximum, then again we transfer the 5,000 liter feed material. So this is how we filled this reactor with 10,000 liters. May I have the next slide? This is the some scanning electron microscopic photograph of the organism that we use. This is Introvector clocki. I told you, IIT-BT0. This is our patented organism, which find very suitable for the hydrogen generation. Then we isolated another bacteria from high oil containing soil, Citrobacter friendly, IIT-BT L139, and another bacteria we isolated from the Swiss sludge, Bacillus coagulans, IIT-BT 81. So all find suitable for the hydrogen production. May I have the next slide? This is the control forward bioreactor that we use. This is the control modules that we have. I told you that, and gas is connected by displacing the this water we can collect here. And the media, initially we use the MOA media, the malt extract, is extract, and glucose. Later on, we replace this with different type of waste material, distillery effluent, starchy wastewater, all these things we use. May I have the next slide? This is the profile of this hydrogen production and cell mass formation. Bottom two line is the cell mass production, and upper two line is the hydrogen production. One is by using glucose as a carbon source, the another is the sucrose is a carbon source we can have. May I have the next slide? And I want to point out that this gas production is so fast that within 11 hours we get the maximum amount of hydrogen production. So our time of fermentation only 11 hours is quite less. And it can use the different type of carbon sources like cellulose, arabinose, fructose, maltose, potato starch, carboxymethyl cellulose, xylose, dextrose, and sucrose. Since it can utilize the different type of carbon sources, so this organism is very much suitable for utilizing the waste material, because waste material also may contain different type of carbon sources. And among these carbon sources, we find sucrose and cellulose is better, best as compared to other carbon sources. May I have the next slide? So this is kind of a comparative table that we have. I told you that we use the Intervector clock A IITBT08, and we also developed double mutant strength Intervector clock A DM11 by closing the ethanol production pathway and lactic acid formation pathway. And by using that, we try to improve that yield of hydrogen, that is 3.8. And we have seen before that a maximum hydrogen yield is 4. So we almost reached to the target. And we try to compare the organism that is used by other group, and we find that our results is quite encouraging. May I have the next slide? So another study, we for the sake of participants, I want to point out that we try to do some kind of metagenomic study and try to find out some unculturable organism, how they are suitable for the hydrogen production. Also, we try to use this organism because I already pointed out that Enterbacter clockis, Citrobacter fringi, and Bacillus bourguignon, they can produce hydrogen in a very good amount. So if you can use this culture, mixed culture, co-culture, mixed culture, if we use, because then how it influences the hydrogen production process we try to observe. Here I want to point out our group established the fact that in the anaerobic digestion process, that particularly in case of the methane fermentation process, that we have two group of microflora. One is called acidogens, and other is called methanogens. So we find the acidogens has the potentiality of producing hydrogen. So so we try to use that acidogenic organism, and we compare that how it is comparable with the pure culture. That we find it is quite attractive. May I have the next? So this is the thermophilic mix consortia we have from the anaerobic digester. We have mesophilic mixed microbial consortia. We have this we can use easily in the in the hydrogen dark fermentation process for converting waste material to hydrogen. Because this is very easy, because as compared to the pure organism, you have to sterile the feed. Am I right? Otherwise your desired organism cannot grow. But when you use the mixed microbial consortia, then this problem, sterility problem, can be reduced to a great extent. And particularly in case of thermophilic mix consortia, we have no problem. And mesophilic mix microbial consortia, we face little problem, not a significant problem we face here. May I have the next slide, please? This is the performance of the reactor. You can see by we using the different type of process, batch process, the regulated pH, mutant strain. We use the immobilized system. We use the different type of organism that is used, and this their how the organism produces the hydrogen. This has been quoted. And you do when you talk about the pure oxygen, pure organism, Enterobacter clocky, we get about 60.9 millimoles of hydrogen per liter, But when you use the co-culture of Enterobacter cloacae and Citrobacter friendly, this increases to 105.4. If you use this plus Bacillus coagulae, it will further increase to 117.4 millimoles per liter. May I have the next slide? And this is the metagenomic concept we try to use in our system. We take the natural microflora and enrich microbial consortia. We prepare that, and from that we use this particular process and try to find out how it affects this hydrogen generation process. So non-culturable that organism that also can contribute for this hydrogen production. We try to compare, but it is not as good as the organism that we use that is reported in case of acidogens of from the anaerobic digester or the Enterobacter cloacae that we isolated. May I have the next slide? This is the different permutation combination of this organism that Enterobacter cloacae, Citrobacter friendly, and the Bacillus coagulant we use in different ratio 1 is to 1 is to 1, 2 is to 1 is to 1, 1 is to 2 is to 1. This, but we find that 1 is to 1 is to 1 give the better performance. Now, here I want to point out when we use this constructed consortia for hydrogen production, we find that it again the growth of a particular organism depend on the substrate because suppose we use the Suez sludge as substrate, then we find when you use this mixed consortia, then we find that Bacillus coagulant they grow their population is more as compared to Enterobacter cloacae and Citrobacter friendly. Suppose when we use the high oil containing soil, then we find that a Citrobacter friendly, that concentration increases as compared to other two bacteria. So they're very substrate specific. When they're since it is isolated from the high oil containing soil, they get the favorable substrate, they will grow more. Like Citrobacter friendly is isolated from the high oil containing soil, and the and Bacillus coagulant it is isolated from the this Swiss sludge. So that is the reason behind that. May I have the next slide? Next slide, please. So different type of waste material can be used. You can see I try to they give you the information that how the waste material coming out from the industry, how it looks. And this causes the health hazard problem. And this is to be utilized and that causes the bioremediation, the environmental health will be taken into account, and this is and that help for the bioenergy generation. It contains the high oil containing, and this liquid, if we allow it to mix with the water stream, it greatly polluted our water stream, and when we drink those water, our health will be affected greatly affected. Some of the waste material that some of the waste like algal biomass DULK can be served as a rich nitrogen and mineral source. So here I want to point out for the students that those who wants to use the waste material as a substrate for the microorganism, they require some kind of nutrient for the growth and multiplication of the organism. So we find that if we use the algal biomass, DULK, or or water hyacinth, if we add that, that increases the growth and metabolic activity of the organism, and that enhances the hydrogen production to a great extent. May I have the next slide? This is a kind of comparison that if we use that glucose as a substrate, how the energy conversion you can calculate if the waste material use as a substrate, how we can calculate the energy recovery from this process. This, yeah, I have shown the calculation. Please check it. And if you have any problem, you can write to me. I shall clarify this. May I have the next slide? So this is the how this gaseous energy recovery takes place if we use the glucose. Enterovector clocky, it is 11.1%. Co-culture of Enterovector clocky, Citrobacter friendly, it is 19.3%. If you use the acidogenic mix consortia, it is 21.4%. So what I want to point out, the acidogenic mix consortia that we isolated from the from the anaerobic digester, we find very useful for the hydrogen production. And this is we established this fact that this is suitable for the hydrogen production. Now, when you consider the distillery effluent as a substrate, waste material as a substrate, we find that that there also their conversion increases. In case of Interbacter clock is 29.1%. In case of co-culture, it is 34.7%. In case of acidogenic mix consortia, it is 42.3%. May I have the next slide, please? This is the system that we develop in our lab. That that we as we have, this is our gas producing system. So if we apply some vacuum, you can remove the hydrogen. Am I right? And we find if you look at the Lee-Catlier principle, Lee-Catlier principle is the Kc equal to concentration of product divided by concentration of substrate. Now, since our product is gas, if you applied vacuum, the hydrogen you can suck. So what will happen? The concentration of the product will reduce in the reactor, and as soon as it's reduced, the more substrate will convert it to hydrogen and carbon dioxide, because Kc should be constant. So that is the so we use the vacuum recycling, and this vacuum recycling logistic control system we develop in our lab. And this find we find that it can improve the hydrogen production to a great extent. And then the corner you can see the solid matrix how it looks, and it shows how the organism are immobilized on the solid surface. Next, I think next slide it will be clear. May I have the next slide? You can see this slide is very clear. In the left hand side you can see the fiber, plain fiber. In the right hand side you can see how the organism grow on the surface of the fiber. At the bottom you can see how the organism is oozes out from the surface of the solid matrix. You can see this is daughter cells. They are coming out from the surface of the solid matrix. You can see it. This is how the process operated. May I have the next slide? So we try to do some kind of material analysis of the process, as you know that, and we considered the basis is 1 kg COD. We find if we use 1 kg COD, we can produce about 80.6 gram of COD. We can convert to hydrogen, but a good, very good amount of that material will remain as a metabolites like acetate, butyrate, ethanol. It can, and also biomass and effluent that will remain. May I have the next slide? And this is kind of a comparison of the different processes that we develop in our lab. We made a comparison. Only I try to, at the bottom I put red mark that you can see that when you use the cane molasses, and that de-oil cake, we find our hydrogen production is about 18.74 moles of hydrogen per kg of COD reduced. The theoretical yield is around 20.6. So this is, we get about 18.74. So we almost reached to the target. So I show you that the performance of my 10,000 liter reactor, you will find that is also quite attractive. May I have the next slide? So this is the what I try to point out. This is the different type of waste material that we have, and we can use some kind of algal biomass is a and the de-oil cake as a supplement, that a nutrient supplement for the cane molasses, distillery effluent, or starchy wastewater. The usually the distillery effluent or starchy wastewater and cane molasses doesn't contain a lot of nitrogen source. They have a lot of carbon source. So they require some kind of nitrogen source for the growth of the microorganism. So this is how we can use, if we mixed in a judicial way, we can use the waste material for the hydrogen production to a great extent. This is the organism that we use for that. May I have the next slide? This is the different de-oil cake we use as a nutrient supplement. One is the groundnut de-oil cake, coconut de-oil cake, mustard de-oil cake, sesame de-oil cake, and rice bran de-oil cake, and soybean de-oil cake. Now we find that groundnut de-oil cake is better as compared to other de-oil cake for this dark fermentation process. May I have the next slide? So as you know, when you operate, this is the characteristics of the organic waste, because whenever we work within kind of waste material, try to analyze that what is the different component present in the system. We analyze with respect to pH, total solid, total suspended solid, total volatile solid, total dissolved solid, total COD, soluble COD, total carbohydrate, total reducing sugar. We try to find out how they are comparable with each other. You find that total solid content is quite high in case of cane molasses as compared to distillery effluent and the starchy wastewater. The COD content also, you see, this is in case of cane molasses very high as compared to this. This is how you can find it out. May I have the next slide, please? This is the different DOL cake. This is the, you can see that 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 it contains the different type of materials. The nitrogen source is one of the very important factor, and they contains very good amount of nitrogen. You can see that in case of groundnut oil, it contains about 6.889. So we find DOL groundnut is very good supplement for the wastewater for the hydrogen generation. May I have the next slide? This is very much essential. You see that how we have utilized this. This is the interactor clocking. When you use cane molasses we use yeast extract as a nutrient supplement, then groundnut dual cake, then distilled dry grain solid DDGS. This we get from the IB agro industry. So if you compare that, you will find that that by using yeast extract we get 1.9, whereas we use the groundnut dual cake we get 2.8. So it is not only the yeast extract is very costly, but the groundnut dual cake is not very, it is a byproduct of the industry, it's not very costly. So this is how we can minimize the feed cost in the hydrogen generation process. Same thing happens to other type of culture. I don't like to repeat it there. Just you can go through the table, you will find it. Only I want to point out in case of thermophilic organism, we use this yeast extract and the tryptophan that is used, and and that is that in the tryptophan is the yeast extract is very costly. That we have to replace with the different type of nutrient supplement to make the process attractive, that economically attractive. May I have next slide? This is the hydrogen production rate that we have in the 20-liter reactor. We get about 67.7 liters of hydrogen per hour. The huge amount of gas is produced. You can see 20-liter reactor, we can produce 67 point, almost about 68 liters of hydrogen we can produce per hour, is quite high, and COD conversion efficiency is 50 to 60%. May I have the next slide? And thermophilic also we get this cane molasses, we get 4.6 liter per liter of the media, and distillery effluent is 2.9 liter per liter of the media, and COD removal efficiency is 60 to 70%. And this operation is carried out in a control fermenter. This is New Brunswick 20-liter fermenter. Now, previous is our custom-made reactor that we have designed by ourselves, and we work on that. May I have the next slide? So this is kind of analysis we get. You can see that with the cane molasses, what is the conversion efficiency and other distillery starchy wastewater. This we try to establish the fact that groundnut dealcoholic is better nutrient supplement as compared to yeast extract and diammonium phosphate, because you know that diammonium phosphate is the chemicals, and yeast extract is kind of that is the required, because it's different biological media we use as a nutrient supplement, but this is very costly material. May I have the next slide? This is our 10 cubic meter plant that we have located at IIT Kharagpur. May I have the next slide? This is how the raw material preparation, raw materials handling is showing here. You can, because we have a 16 minutes video that is available in our website, that is the Bio H2 IIT KGP. So if you are interested, please see the website and see the video. You can find that how we operated the plant. You can have the idea. May I have the next slide, please? Or I can request Professor Pandey, Anjana Pandey, to show the flight in the in some of the presentations, so that that all the audience, all the participants can see how that 10,000 liter reactor operated at IIT Kharagpur.
I will do it, sir, definitely.
Acha. Okay, that will be very great. Huh? This is this you see that in the performance of 10 cubic meter reactor, our maximum production is 76.2 cubic meter hydrogen per day that we produce. That this small plant that can give, because this hydrogen, this gas doesn't contain carbon monoxide or H2S or sulfur dioxide. So it is no, it is this gas can be easily used in the fuel cell as a fuel, and convert, you can convert, if it passes through the fuel cell, it is easily converted to electricity. And we have calculated that if we produce electricity, it can give light to a small village. So you know that this just a small plant is good enough to give a plant light to a small village. So if you increase the size and industrial level, you see that how much benefit we can get out of that. May I have that? And I want to point out, as the present circumstances, that all the distillery industry, that 50% of their that energy they use for the distillation of alcohol, that they meet from the methane that produced from the anaerobic digester. So if we add that hydrogen in it, then naturally that utility of the process will increase. Another thing I want to point out, that that biohythane, hythane is very important, because I have already pointed out that methane is the lowest hydrocarbon, is used as a CNG for the operation of different three-wheeler, four-wheeler. They did, and it poses little that pollution problem and fogging problem can be removed. Now we find that the fuel efficiency of the process can be improved if we add 7.2% of hydrogen with the methane. So we call hythane. So, but since one reactor will be producing hydrogen, another reactor hydrogen, another reactor methane. So, if we mix 7% of hydrogen with methane, this improve the efficiency of the fuel to a great extent. Now, this is the scale up concept that we have. You see the cumulative hydrogen production, 0.002 cubic meter reactor is 2.6 per liter of liquid. And then as we go for higher scale, zero point zero 0.05 cubic meter is 2.9, and 10 cubic meter is 7.6. So scale up, if we go to the higher scale, we observe our our hydrogen production increases to a great extent, because there the contamination problem is comparatively less. And since a lot of gas production is there, there is a kind of gas causes some kind of hesitation in the system that improves the efficiency of the process to a great extent. May I have the next slide, please? This is the calculation we made, and we find that about 37.9% energy can be recovered by using this hydrogen production process. And hydrogen production process that then if we and I have already shown you that that it produces a lot of acetic acid and butyric acid and ethanol, which is a good substrate for the methanogens. Then we pass through the methanogenic reactor, we can produce methane and carbon dioxide. So total energy recovery will be more than 60-65%, and that is really attractive. So if we go this process, then really that we'll find that our biological energy generation process will be more they're quite feasible, quite efficient for the energy production. May I have the next slide, please? So this is the comparative study of this, that we have different type of pilot plant has been carried out, and we find that this our as I pointed out, our production is 18.74 moles per kg of COD reduced. And but you know that rain atoll that are reporting as 26.113 moles of hydrogen per kg of CO2 removed. So we actually theoretical amount is 20.6. I don't know how he is reporting 26.13. This might be some miscalculation is there. Otherwise, as compared to the other researcher, our results is quite encouraging. May I have the next slide, please? So this is the my research group who contributed in the research work, because we have this is several persons they contributed in the research work. And this is the last phase, because these are the research work fellows that were involved with me for finishing our hydrogen the research at IIT Kharagpur. And may I have the next slide? So this is the first book we have written. This is biohydrogen production. This published in the year 2014. Then this is followed by biohydrogen process. And this biohydrogen book is the first in the world, because nobody has a reported book on biohydrogen. So we first wrote this book. And may I have the next slide? Then 2020, we have written Fundamentals of Biofuel Production Process, where we discuss all the biofuel production process in details. So this year we published the Industrial Biotechnology book. I think those who are interested in the biological field or biotechnology field, this book will be very, very useful for them. Please consult this. Both the book published by CRC Press. May I have the next slide? Besides that, I edited two books. One is the Algal Biorefinery and Integrated Approach, and there is Microbial Fuel Cell. And that it converts waste to what actually. That is how the electricity conversion take place from waste to what. So this published by the Spinger. So I think this can enrich your knowledge on biofuel production processes, and I'll be very happy to answer any question if you have. Or today I find that time is already up. So if you have any question, you can write me, or you can write to the program coordinator, Professor Pandey. If she pass it to me, I try to answer those questions. With this, I thank you very much for your kind attention, and I hope that young people should come forward to make the biofuel production process more successful in the years to come. Thank you very much.
Thank you, Professor Das, for your wonderful and in depth explanation on scaling up biofuel production using organic waste. I hope our participants enjoyed the session thoroughly and will get benefited. Dear participants, the session is now open for discussion. Kindly put your questions forward. As I can see, Mamta Gautam has some query that needs to be addressed. Ms. Mamta, please proceed ahead.
Mamata, can you tell me what is your question? Or you can write in the chat box. I try to answer.
Any other participants that wants to ask some question to sir? Please go ahead. Participants, do you want to ask anything? Dear participants, you want to
Hello,
ask anything? Yes, ma'am. Please go ahead.
am I audible?
Yes,
Yes,
yes.
ma'am.
Sorry, I disconnected in between the talk.
Okay.
Hello, hello,
Yes,
am
yes.
I
No
audible?
issues, ma'am. Please go
Yes.
ahead with your query.
Okay, thank you, sir, for a nice presentation.
Thank
So,
you.
I would like to know what is the future of this technology because on the lab scale it is successful. But what about the field application of this?
No, I think this is a very
Biofuel production, I'm talking about biofuel production from
okay,
algae
okay,
and
okay,
from
okay, okay.
bio
I'm covering all. Okay.
Okay.
Now, as per methane production process is concerned, that is already successful throughout the world, both in the small scale, that is, as well as in the large scale. So I can find that in the Western country, they operated buses by using biogas, because and CNG is largely used throughout the world for reducing the environmental pollution problem. So as per me, the anaerobic digestion process, that is not a problem. Now, major issue that comes with the hydrogen production, because this is the new process that is coming up. Now, as I pointed out, we are working, because this process presently we find that electrolysis process are very much attractive as compared to other processes that we have. That is why government of India, that they are giving more stress on that. Now, biological hydrogen production, as I pointed out, that we have been working with a strong group, with the 5 different groups in India, and try to find out how it is suitable for the commercialization of the process. That is why we have come up with the performance of the pilot 10,000 liter pilot plan. Now, our process has taken by the industry to go for the commercialization. So until and unless we have data from them, that we cannot see that commercial viability of the process. So we are desperately looking for for that, and as I pointed out, that the hydrogen production alone will not be attractive. The hydrogen, biohydrogen followed by biomethane, that is will be more attractive, and that will be positively there in the years to come. Just you wait 1 or 2 years, you will find a lot of industry, lot of already I find a lot of different industry, they are working on this process. This process will come very easily. Now, as far biodiesel production is concerned, that a major issue is that algal biomass production and the algal biomass separation. But the major cost involvement is there. So you have to find out high fat containing algal biomass. Then you have to increase, because if you want to increase the fat content in the algal biomass, you have to we find the growth of the algae that reduces drastically. So you have to find out some kind of strain which can higher growth and higher fat content. Then you have to find out the reduction of the algal biomass separation process, because algal biomass concentration is very less in the liquid that is to be concentrated, that is to be separated, and that separation cost about 30%. So if we can reduce, then it will be very attractive. Then we find this fat is very good for as the biodiesel. The reason is that it contains good amount of unsaturated fatty acid, and due to the unsaturation, even in the cold country, this diesel will not be solidified. So in that way, this is we have work on that, and please consult our book with that we have written. We already established that this process will be, and as per but algae can be used for other purpose also. There are different organization in India and abroad. They are using algae for other as a feed, and feed for the man as well as for the animals. They're different, and also use it has a lot of medicinal value also. Now let me come to the bioethanol production process. Bioethanol production from the cane molasses, that is already largely in operation in India, because our alcohol is mostly produced from cane molasses. In the Western country, it is produced from beet molasses, and some of the industry in our country produces alcohol from the starchy material like broken rice and all these things. That is already successful. Only this thing is that they are trying to use the lignocellulosic material, and the lignocellulosic material is not very much successful, because the reason is that the removal of the lignin, then the degradation of the cellulose. I told you, the removal of lignin is a problem, and after removal of lignin, the kind of cellulose present, that is, if it is the crystalline cellulose, which is very difficult to degrade, the naturally that you have to find out the kind of technique how it can be easily degraded and produce the soluble carbohydrate, so that we can easily convert it to ethanol. This is so cellulose to lignocellulosic material to ethanol. So though Prague, they are reporting that in India they are reporting they are successful for producing in a commercial scale, but I have a lot of doubt on this, because the enzyme that is responsible for the degradation, that should be quite economic. Otherwise, the cost of the production process cannot meet with the cost of production process from cane molasses or starchy material. Is it clear?
Thank
Hello?
you, sir. Thank you for explaining. Hello.
Uh,
Sir, I'm Dr. Shweta Kotesh from MIT University, Rajasthan.
okay.
Sir, thank you for explaining each and every aspect related to biofuel production. What are the limitation, and how can we start, and it can be established in 1 or 2 years. There are many companies as per the information you are giving to me. So I'm very much interested in this. I have several more queries. I will talk to you regarding this. I would like to know your email address so that I
That
can contact you.
you go to my that my website already given. In the website,
Yeah,
everything
yeah, yeah,
details is given. You go to the
yeah, yeah,
website, you will get the all the information in details.
yeah, yeah, yeah. Yeah. Thank you, sir. Thank you for a nice
Thank you
presentation
for that. I want that young generation
and
should come.
enhancing my knowledge regarding this, because every time I would like to think what is the feasibility of the technology. What, yeah,
Okay,
we,
okay.
right. Thank
Thank
you,
you
sir.
for your good question actually.
Thank you, sir.
Okay, sir. Okay, sir. There's one more question from the participant. How
Yes.
can I reduce free fatty acids? What are the techniques that need to be performed to reduce
Okay,
from the
and
reduction?
that how we can reduce? What is the question?
Sir,
Hello,
how
can you repeat?
can how can we reduce free fatty acids, and what are the techniques required for the reduction?
No, free fatty acid from where?
Miss Miss
Free
Prabha,
fatty
are you there? You can ask your question directly to sir.
free fatty acid from where? Because that is to be now free. If you talk about the hydrogen production process, because already the free fatty acid will be formed because volatile fatty acid like acetic acid, propionic acid, butyric acid. Now, in case of the biodiesel production process, that we in the fat is usually considered as ester. So ester, this undergo the trans-esterification process, both chemically and enzymatically, and which produces monomethyl ester, and that we call it call it as that biodiesel. And now free fatty acid concentration, if you consider as in the biodiesel production, that you have to go in the process in details because it varies from the process to process actually. And as per process is concerned, process free fatty acid will not be there. Only during the trans-esterification process, there is a possibility of presence of some free fatty acid.
Thank you, sir. Any other query, participants? If there's no query, then we'll be closing the session for today.
Thank you very much for your attention, and I'll be here. I am not moving out. So, if anybody need any kind of clarification, please let me know. So, I'll be very happy to give the clarification. Okay.
Thank you so much, sir.
Thank you.
Everyone has liked your presentation and the way of presentation.
Thank you. Thank you very
They are
much.
very much curious to learn
I'm
this thing.
thankful. I am thankful to you also to give me an opportunity to
It's my
share my
pleasure,
ideas.
sir. It has always been my pleasure. Thank
Yeah.
you so much.
Thank you. Should I leave
Announcement
now?
करो.
Sir,
Sir,
please,
please, sir,
you
you
may
may
leave.
leave, sir.
Okay. Okay. Thank
Thank
you.
you, sir.
तुम कुछ बोलने वाले?
Dear participants, we are closing for this today's session. Tomorrow at 10 a.m. we'll be starting our third session. Kindly join 10 minutes before. The link will be provided to you again.
क्या भेजा है?
Thank you,
So
sir.
we are done for today. Thank you, everyone. You may kindly leave, and we'll see you soon tomorrow at 10 a.m. for the next session.
Ma'am, time, what time for tomorrow?
So, third session will be starting from 10 a.m. and the participants need to join 10 minutes before.
10 minutes ago. Okay, ma'am.
Yes, sir.
Okay, ma'am. Thank you very much. Thank you, ma'am. Thank you.
Thank you. Thank you. Welcome.